Hey everybody, I'm back at you with a little bit of a annotated PowerPoint lecture. In this case, we're going to cover meiosis. So I hope everyone's doing well, and I hope everyone's ready to get back into kind of the new normal swing of things. I know I'm excited to kind of get back and get back to the content. So without further ado, we're going to talk about meiosis today. And the relevant textbook reading for this section is really just one section of the textbook. It's a pretty meaty one, so it's pages 103 to 110. This is section 4.3 of chapter 4, so kind of a follow-up of the mitosis stuff. And so kind of moving into this lecture, one of the things that I want you to have already kind of learned, or what I want you to already know, is broadly what occurs during each phase of meiosis. So this is generally the same philosophy I had going into the mitosis, the lectures that I posted last week. So I want you to know what's happening during prophase, metaphase, anaphase, telophase 1 and 2 of the two different phases, larger phases of meiosis. And then additionally, I want you to be familiar with this idea of interkinesis and how that's different than cytokinesis. So let's get into it a little bit. So I want to start with just a little bit of a review of mitosis and kind of what we think about that one. So mitosis is relatively simple, especially compared to meiosis. So in this case, mitosis, sorry, I'm going to hopefully not confuse those two as we move along. So mitosis starts with a single mother cell. Okay, and then the end product of mitosis are two daughter cells. Okay, so the key with mitosis is that these daughter cells are genetically identical. And that's kind of one of the big ways that mitosis differs from meiosis. Okay, and so this mother cell, as we've kind of covered a little bit earlier, this really kind of starts after duplication. So the mother cell really starts with the genomic content at 4N. So if 2N is diploid, this was, I guess, quadruploid, but the sort of normal diploid genome has divided. And so then when this gets divided equally between the two daughter cells, each of the two daughter cells then end up as diploid cells. Okay. But the key here is that they're identical. We haven't had any real change in the DNA content between these two things. Okay, and so the key is that mitosis occurs in somatic cells. So these are most of the cells in your body. Skin cells, muscle cells, and intestines, even neurons are examples of somatic cells. So when they divide, they're really just proliferating to make more cells, and there's no real sort of introduction of genetic variability within those cells. And so I think you can really think of this as it's maybe not the inheritance that we think of classically, but I think it's okay to think of this as a form of cellular inheritance. Okay, so for example, in this mother cell, if we just take maybe one kind of duplicated chromosome here, so each of these are sister chromatids. And so this kind of relates back to a question that I got maybe a couple weeks ago, or maybe 3 weeks ago now. I think it was from Courtney asking about what, this idea of inheritance of mutations. And so you could imagine a situation where if this mother cell actually incurs some kind of mutation in the DNA, that these daughter cells could actually inherit these mutations. And now this is not the kind of classical form of inheritance that we'll end up talking about kind of starting later this week. But this, I think, it's fair to think of this as a form of cellular inheritance. Okay, so that's mitosis. So let's move on and give a broad overview of meiosis in this case. Okay, and so meiosis similarly is going to start with one cell. In this case, meiosis starts with germ cells. But the end product out of meiosis are 4 cells. Okay. Now, hopefully you're familiar with what these are. I'm sorry, these are roughly circular. But these 4 cells that come out of meiosis are known as gametes. Okay. Now, meiosis, one of the big ways that it's different from mitosis is that this occurs actually in 2 rounds of cell division. So there's going to be some kind of intermediate cell type before we get to these final gametes. Okay, so this first germ cell will undergo 1 round of meiosis. So this is meiosis 1. And then these intermediate cells will then undergo a second round of meiosis to generate the gametes, and this is known as meiosis 2. Okay? And so similarly to mitosis, this germ cell starts after DNA has been replicated. Okay, so it's now at a genomic content of 4N. But then when all is said and done, although there's two rounds of through these 2 rounds of cell division, and these final gametes end up with a genomic content of N, meaning that they are all haploid. Okay, so this is really the goal of meiosis, is to generate haploid gametes. Okay, so it's tempting to think that these cells here in this intermediate would be 2N, but that's not actually the case. As we'll see, their genomic content isn't really diploid anymore. They've actually undergone a reduction in the total number of chromosomes, even though the sort of chromatids makes it seem like it isn't. So these aren't really diploid in here, but we go from this kind of 4N content all the way to a haploid gametes. And so again, the key with meiosis is that this occurs in germ cells, and this and then is able to then produce gametes out of these germ cells. And so this is really kind of a quintessential example of organismal inheritance. This is how it happens. Oops. Sorry about that. Okay. So this is really how genes are passed down in between generations. This is really the function of germ cells and gametes. This is what they do. Germ cells are cells that specialize in the generation of gametes, and gametes are the cells that actually pass genes down between generations. So that's the overview of meiosis. So let's get into some of the particulars. But before we get there, just a broad way to look at sort of why meiosis. This is just kind of reinforcing some of the things I've said before, just in a different way. So again, if the endpoint of meiosis is to take a single germ cell and generate haploid gametes, N. Again, these are all haploid gametes. These gametes are then important because they then form the basis of fertilization. So if we take one of these haploid gametes over here, it can then combine with other gametes from other organisms to then allow for fertilization and the formation of diploid cells. Okay? So again, we go from this initial genomic content of 4N to make these haploid gametes. Haploid gametes can then combine during fertilization to make a final diploid organism. And in the process, what they're doing is they're combining genes from different sources. So again, this is another way to increase genetic variability between generations. So that's just kind of broadly why meiosis is important. Okay. So let's focus in on meiosis a bit, and we're going to focus in on prophase 1. Okay, so I'm going to contrast this with prophase in mitosis, and in fact, this is going to look a little bit more like prometaphase. So if we think back to some of the hallmarks of prometaphase in mitosis, we have our single mother cell here. At this point, the nuclear envelope is broken down. I'm not going to draw them in here, but the centrosomes have moved to each pole of the cell. Each of the ends or the poles of the cell. And the chromosomes in this case have condensed. We can see them. And so I'm going to go ahead and draw in here two duplicated chromosomes. Okay? So we'll call this one chromosome 1 in black. So it has duplicated, and we'll say that this is sort of one version of it, and let's say the homologue in the dotted line in black has also duplicated. Okay, so we can actually see that. And let's sort of, for reference, let's throw in another smaller chromosome here, another one that has condensed, so we can see it. And here's the homologue that is also condensed, and I'm just doing that in dotted lines just so we can differentiate the two homologues. Okay? So one thing you want to kind of notice here is that these homologues in mitotic prophase or prometaphase, they don't pair up. Okay, so they're existing really separately, and that's going to be a big contrast between mitotic prophase and what's going on in meiosis in prophase 1. Okay, so I'll just put a little dotted line here. So in meiotic prophase 1, it looks substantially different. Okay, so if I draw the cell out here, and we'll have an example, again, the genomic content of this mother cell in mitosis is 4N. We have the same 4N genomic content in meiosis because the genome has replicated. Okay? And so if we have our two sets of homologs, they look a little bit different. Okay? So in this case, here's the homolog of this chromosome, and they actually pair up with the other homolog. Okay, so there's that one there, and we'll draw the red one down here. Okay. So there are cellular mechanisms in place that allow these two homologs to recognize each other and exist sort of right next to each other. And this is where we get the term then homologous pair. Because the homologs for each chromosome pair with each other. Okay, so this here is one homologous pair, and this red down here is another homologous pair. Okay, so what's actually kind of happening in this homologous pair? How are they attaching to each other? So I'm going to zoom in on this homologous pair up here, and I'm just going to draw them in a little bit of a different way. So here we've got the one homolog here up on top. And I'll draw the other homolog down here, and dotted lines. Okay, and so the reality is that these are actually physically attached. But just first, before we get there, a technical term. This whole thing here is what's known as a tetrad, or you also see it referred to as a bivalent. Tetrad because there's actually 4 different chromosomes here. So you've got, in this case, 2 sister chromatids of this chromosome and 2 sister chromatids of this one. So we've got 4 total chromatids which make up this tetrad. Okay, and so as I mentioned, these things are actually attached to each other. Okay, so I'm drawing these kind of small dotted lines in here, but there's actually a proteinaceous kind of complex and structure in between these 2 non-sister chromatids. And this is what's known as the synaptonemal complex. Okay, so in future lectures, if I refer to this, let's go back. I refer to this, I'll probably abbreviate this as SC, just because I don't want to keep writing out synaptonemal complex. But this is a synaptonemal complex, so it's a proteinaceous complex that exists in between these 2 non-sister chromatids and allows this homologous pair to undergo what's known as synapsis. Okay, so the process is of these homologous pairs attaching to each other is known as synapsis, and this happens by way of the synaptonemal complex that exists in between these 2 homologous chromosomes to then create the overall structure called a tetrad. Okay. So this is all fine and good, but there's actually a lot of really interesting things that can happen because of the arrangement of the homologous pair like this. And so this brings us to really one of the key aspects of meiosis, and this is crossing over, hopefully some term you've heard of before. And so we're going to get into that in a little bit of detail here. So this again occurs during prophase 1. Okay, again, a lot of actions happening during meiotic prophase 1. Okay, so I'm going to zoom in on one of these tetrads. Okay, so if we've got one homolog up here. Okay, and just for kind of reference's sake, I'm no longer going to draw these in dotted lines. I'll just draw the other homolog of this pair in another color, in red in this case. Okay. Okay, so these are kind of right next to each other. Okay, and then they are attached via the synaptonemal complex between non-sister chromatids. Okay. Now what happens in prophase 1 of meiosis is that these actually undergo a process known as crossing over. Okay. and the chromosome, the chromosome arms, basically the chromatids, actually physically cross over with each other. Okay, so there's one of the homologs up there, and the other one is down here, and you can see that I've drawn that the non-sister chromatids actually cross over with each other. Now keep in mind that these things are still attached. I'm just kind of drawing them separately, but they're still attached via the synaptonemal complex here. Okay. But then these points, right here and here, are actually crossover points. So these are important because these crossover points, non-sister chromatids, are actually going to exchange DNA. Okay, they're going to swap some DNA. And this is a process they're actually going to recombine, which is why this process of crossing over is often known as recombination. Mixing things up a little bit. Okay, and so another important term, these points of crossover here and here are known as chiasmata. Okay. So a key concept that I'm not going to get into any detail here, and I think I want to talk about in future lectures, is the idea that this recombination between non-sister chromatids at these points of crossover occurs because of sequence similarity between non-sister chromatids. So remember, these are homologous pairs of chromosomes, which means that in large part, the DNA sequence is very similar. Now, there are differences. They do maybe carry different alleles of different genes, but by and large, the sequence is very similar. And so this recombination, where they can actually exchange DNA, results because of that sequence similarity. Okay? And so we're going to, probably in a future video, we're going to talk about kind of how this is happening at a more molecular level. And so this is going to be in Chapter 6, page 203 to 207, the section entitled Crossing Over at the Molecular Level of Model. There's actually some really interesting molecular biology that underlies how these non-sister chromatids are actually able to exchange DNA. Okay? So let's dive into this, kind of keep diving into this concept of recombination and really kind of illustrate then what is the consequence of this recombination. Okay, so here I'm going to draw out, we're going to follow two hypothetical genes in this case. Okay, so gene 1 and gene 2, and so gene 1 has kind of a capital A and a lowercase a alleles. And then gene 2 is going to be denoted, it's got two different alleles for its gene, capital B and lowercase b. Okay, so these are the different alleles or versions of the genes that we're going to track on these chromosomes. All right, so if we look up here, and we're going to have maybe one of our homologs up here. We'll say in this case, we're just going to focus here on this left chromosome arm, and we'll say that in this case, this chromosome is homozygous for each of these alleles, but in this case, it's homozygous for the capital A and homozygous for the lowercase b. Okay? And then we're going to contrast that with another chromosome, the other homolog down here. I'll draw them out just a little bit separately to make it clear. But again, these are undergoing synapsis. I'm just not drawing it then anymore. And so in this case, this chromosome is homozygous for the lowercase a and homozygous for the capital B, just in terms of our hypothetical situation. Okay? So let's then presume that during prophase 1 of meiosis, this homologous pair undergoes crossover at a point in between the A and B alleles on this chromosome. Okay, so let's kind of walk through what the consequences are for this recombination. So if we actually get an exchange of DNA between these two chromosome arms, we end up with some different looking chromosomes. Okay, so the kind of top chromatid of this chromosome actually stays the same, but this bottom one then is going to end up carrying a little bit of the DNA from the other homolog that I'll draw in red. Okay. Likewise, if we look at this bottom chromosome, this bottom chromatid will be the same, but this top chromatid will carry a little bit of DNA from the other homolog. Okay? So there's been an actual physical exchange of DNA through recombination. So where this becomes important is actually we can see this is important when we track what's happening to the actual alleles on this chromosome. Remember, genes reside on chromosomes, and that's how genes are passed down between generations. And so here, this top one still has a capital A, a lowercase b, still has a lowercase b here. But in this case, the other capital A or uppercase A allele has actually been moved down to this other homolog. Okay, and if we look at the alleles on the red homolog, so in this case, we still have capital B, those haven't moved. This bottom one still encodes for the lowercase a allele, but that lowercase a allele for the other one is now moved up here. Okay, so if we look at then the genotypes present on each of these different chromosomes, the genotypes for both these chromosomes is uppercase a, lowercase b. And the genotype for both of these is lowercase a, uppercase b. Okay? But things get a little bit different when we start to look at the genotypes encoded on each of these different chromatids. Okay? So in this case, the top one is still uppercase a, lowercase b, and this bottom one down here is still lowercase a, uppercase b. Those are identical to the genotypes that we see. We'll call these the parental genotypes. Whereas now on these chromatids that have undergone recombination, we see some novel genotypes. So this is now lowercase a, lowercase b, and we've got uppercase a, uppercase b. Okay, so these are, oops, kind of messed that one up here, but these are now new genotypes. We'll get to this a little bit later, but these are now known as recombinant genotypes. Okay. So basically, recombination in meiosis ensures that none of the gametes are genetically identical. So each of these 4 genotypes will then be present in each of the 4 gametes generated from this germ cell during meiosis. So out of one germ cell, we can then generate 4 really non-identical genotypes. So that's one of the big keys in recombination. And so this is a very key source of a genetic variation as genes are passed down from generation to generation. All right, so let's kind of walk through kind of broadly then what this looks like through metaphase, anaphase, telophase, and then interkinesis. So just a key term here, interkinesis, is that period in between meiosis 1 and meiosis 2. And it can kind of vary in length, but it's different than cytokinesis. Okay, so let's take a look at what's happening then during metaphase, during meiosis 1. And so in this case, in metaphase, the chromosomes have condensed, they've paired up, they've undergone recombination, but they're still attached, and they're going to then align, just as they do in mitosis, along the metaphase plate. Okay, so we'll have 2 homologs here, one in black, and I'll draw the other one in red. Okay, and so they're undergoing, or they've undergone crossing over, but they're still attached. So at this point in metaphase, synaptonemal complex is dissolved, but these 2 chromosomes are still attached at the chiasmata, those structures that still exist at the crossover points. So they're still held together at those chiasmata while these homologous pairs are aligning along that metaphase plate. So this is really kind of similar to that spindle checkpoint that we talked about in mitosis, where the cells need to really ensure that these chromosomes are aligned to the metaphase plate before they undergo then the next phase, which is anaphase. Okay? And so in anaphase of meiosis 1, instead of sister chromatids pulling apart, homologous pairs pull apart. Okay, so that's distinctly different than what happens in mitosis. So in this case, you've got a homologous pair heading this direction, and so it's carrying with it both sister chromatids because they're attached. And then remember, this one has actually undergone recombination, so it's pulling with it a little bit of DNA from the other chromatid. Okay. Likewise, over here, this chromosome is being pulled to the other end of the cell. And it also, because it underwent recombination, now is pulling on a little bit of different DNA with it to this other end of the cell. So that's happening during anaphase. Okay, and then the last two steps of meiosis 1 are telophase and interkinesis. Okay, and so at this point then we're left with a single homolog in each of the final cells. But remember, there's been a little bit of recombination, so we've actually introduced some genetic variability into the system. Oops, kind of missed there. Sorry about that. Okay. And so kind of going back to this whole process, again, this is sort of, you can think of this as being the homolog that's derived from the mother, and maybe this red one is the homolog derived from the father. And so now sort of the maternal and paternal DNA that existed in this germ cell are being kind of separated out. Okay. All right. So then, kind of to sum this up then, we'll look and see what's happening during meiosis 2. So the big difference here is that at the end of meiosis 2 is cytokinesis, not interkinesis. Okay, and so this whole thing starts then we're in the metaphase, so things are going to align along this metaphase plate. Now what's going to align then is going to be one of these homologs. So the pairs have already broken apart, so we've only left with one. And in this case, we're going to focus in on, we'll just follow from this last slide, we'll follow this one right here. Okay, so this one actually is carrying along a little bit of the DNA from the other one. And so these are aligning on the metaphase plate. And now things look a lot like mitosis. Okay, so in then anaphase, sister chromatids are going to pull apart. So each of these are sister chromatids here on the left and the right. And so in anaphase then, sister chromatids are going to pull apart. So you got one heading this direction and the other one heading this direction. Now remember, this chromatid underwent recombination, so it's got a little bit of a different genetic makeup than the other chromatid. They're not identical anymore, a big difference between meiosis and mitosis. Okay, and then during telophase and then cytokinesis, these two chromatids end up in separate gametes. Okay, so we've now finally generated gametes out of this. Okay, so I'm only showing two here, but remember there are two of these intermediate cells. So that we can generate a total of 4 haploid gametes. And again, these gametes are no longer genetically identical because they've undergone recombination. Okay, so last little concept that I want to cover here is this idea of non-disjunctions. You'll oftentimes hear this referred to as chromosomal non-disjunction. Okay, and so this is actually a pretty big problem for cells if this occurs. Okay, and so non-disjunction, the definition really is the failure of either homologous pairs from separating during meiosis 1 or of sister chromatids from separating during meiosis 2. Okay, so there's 2 times during meiosis that non-disjunction can occur. Each time it's problematic. Okay, so let's kind of diagram out what this looks like in meiosis 1. So in meiosis 1, if we've got a homologous pair, kind of messed that one up. I'll redraw that. And so here I'm just going to draw the 2 pairs in the same black color just for simplicity's sake. Okay, so if they're aligning here, let's say this is during metaphase, and then as things divide going through meiosis 1, if this cell then, or if this chromosome undergoes non-disjunction, remember there are other chromosomes present here, we're just focusing on this one, if this chromosome undergoes non-disjunction, then both members of the homologous pair, both homologs, end up in one of the cells, these intermediate cells, and this other one doesn't get any. Okay, so you can imagine that's pretty big problem for this cell. So in this case, the two gametes that are derived from this cell during meiosis 2 each will have 2 copies of the chromosome. And then the two gametes down here will have 0 copies. Okay, so right away you can start to imagine how much of a problem this might be. So twice the number of genes in this case, and none of the genes in this case. So that's a pretty big problem. Okay, let's contrast that with what's happening, or what this looks like in meiosis 2. So in meiosis 2, again we're starting with just a single homolog, and then meiosis 2, sister chromatids from that homolog separate. So in this case, if non-disjunction occurs, both chromatids end up in one cell, and neither of the chromatids end up in the other one, which is, again, just as problematic. So in these gametes up here, there are twice the number of genes present because there's 2 chromosomes there, and then here there are 0 down here. Okay, so that's very much problematic. Now, the kind of big thing I want you to remember with non-disjunction is that this is often, but not always, lethal. Okay. So because each of these chromosomes contains thousands of genes, we're talking about a massive change in really the genetic makeup of the gametes that arise during this process. Okay, and that kind of change in the genetic composition of a cell is very, very problematic. So you're not just changing one gene, you're changing thousands of genes, and oftentimes this is lethal. Now there are exceptions to this, and most of the exceptions to this occur as it comes to non-disjunction of the sex chromosomes, and we'll have some specific examples of that coming up in future lectures and future problem sets. Okay, so again, this is often but not always a lethal problem. Okay, so I want to finish things up with this lecture, and I want to finish up with a discussion question that I'm going to post on Katie that I want you guys to work on in your small groups. And I'm going to keep some tabs on this, and I want to make sure that you guys are actually doing some of this work. Okay, so here's the discussion question. We've talked a lot today about synapsis and recombination between chromosomes. So we've kind of kept it general, and I've really kind of referred to these hypothetical autosomes. But as it turns out, synapsis and recombination occur between the X and Y chromosome, and this is in fact required during meiosis in males. Okay? So given that the X and Y chromosome vary in their overall structure and DNA sequence, how does recombination take place between the X and Y chromosome? Okay, I made a big point earlier during this lecture of talking about how this crossover occurs because of sequence similarity. Okay, so I want you to actually look into this, and you may need to actually get on the internet and poke around a little bit to find an answer to this one. And then as a follow-up to this question, I want you to kind of take this one step further. I want to try and bring this back to something we've talked about before, which is this method called FISH, or fluorescence in situ hybridization. So as a follow-up to this question, then, how could you employ the technique FISH to demonstrate how recombination occurs between the X and Y chromosome? Or how can you really use FISH to show that this might be a thing how this might happen? Okay. All right, last thing, one more 90s movie recommendation for the week. So this one goes all the way back to 1992. This is one of my favorite movies growing up. Again, not to date myself too much, but this is the movie called White Men Can't Jump, starring Wesley Snipes and Woody Harrelson back when they were much younger. Wesley Snipes before he got totally juiced up on steroids, and Woody Harrelson back when he still had hair. Kind of a fun sports comedy type movie. Yeah, it's a fun one. Give it a read or give it a watch if you get the chance. All right. I'll hopefully talk to many of you in our Zoom online meetings this week. Take care.